Speciation is the origin of new species, and it's a focal point of evolutionary theory because evolutionary theory must be able to explain how new species originate and how the population evolved. Reproductive isolation is a way that we can keep species separate, and so there's two types: prezygotic barriers, which prevent the formation of the zygote or egg, and examples of those are habitat isolation or behavioral isolation. And then post-zygotic barriers prevent the development of viable or fertile offspring, and an example of this is reduced hybrid viability. So there's two types of speciation. Allopatric is when there's a physical barrier that's dividing two populations, whereas sympatric is where there is no physical barrier, but you get these development of different species through either something like duplicated chromosomes or ecological isolation. So phylogenetic trees are a really useful tool because they allow us to track the evolution of species over time. And so the bottom of your tree will be your ancestral species, and then the species at the top of the tree will be your present-day species. And each branch point actually serves as a point of speciation, and each branch can serve as a new trait that might have appeared in that new species. Clades include a common ancestor and all of its descendants. And as you can see from this diagram, all clades are actually nested within a larger one. And clades are actually the same as monophyletic groups because, again, they include the common ancestor and all of its descendants. So polyphyletic groups are another type of group that you'll see in phylogenetic trees and that you might be asked to identify. And so these are groups that have independent origins and no common ancestor. So if you look to the diagram to the right, the Mammalia and the Aves genus share no common ancestor, and so that's why they're polyphyletic groups. The final type of group that you might be asked to identify on a phylogenetic tree are paraphyletic groups. And so these are groups where some, but not all descendants are from the same common ancestor. And so the easiest way I like to think about it is just think about a monophyletic group, but just remove a couple of the species within that monophyletic group. So now shifting gears to talking about the origins of life. So the early environment of the Earth was actually very different from what it is now, in that it had very low oxygen levels. And so some bacteria early on evolved photosynthesis, which allowed oxygen to accumulate in the atmosphere, and this eventually allowed for the evolution of ATP synthesis, which is an oxygen-dependent process, and this ultimately increased the complexity of life. A key theme of this class is that evolution is a tinkerer, and what this means is that instead of making a bunch of new processes, evolution actually fixes existing ones to make them more efficient. And so two examples of this are that the pathways used in ATP synthesis are at least partially conserved from photosynthesis, and that photosynthesis itself is actually partly borrowed from anaerobic processes. Although we see predominantly land plants today, plants actually originated in the water, and this was for good reason. Water allowed for easy transport of gametes, it provided a lot of nutrients to the plants, and it also prevented the plant from desiccating. So if life was so good in the water, then why did plants want to move onto the land? For one, there's more direct sunlight on land, which allowed for more efficient photosynthesis. And related to that, there's also better access to carbon dioxide on land. So both of these help with photosynthesis. There's also a lot more nutrients and minerals on land. And also there was initially the absence of herbivores. So plants didn't really have a predator on land. So naturally, plants had to make a lot of adaptations once they were on the land. One was that they had to conserve water, and so they evolved a waxy cuticle. They also evolved a vascular system so that they could transport nutrients against gravity. They also had to develop new reproductive structures for gamete transfer, and along with this, they also had to protect or reduce their vulnerable life stages, which led to the development of the sporophyte and the gametophyte. The diagram to the right shows the alternation of generations of the plant life cycle. So if we start up top at the sporophyte generation, that undergoes meiosis to produce haploid spores, which develop into the gametophyte generation, And that generation undergoes mitosis to produce gametes. And when those gametes fuse together, they make a two-end zygote, which then develops again into the sporophyte generation, and the cycle continues. Just as the Cambrian radiation led to the mass diversification of animals, there was also a land plant radiation. The first land plants that evolved were non-vascular, which led to the evolution of vascular plants, such as ferns. And then eventually we saw the first seed plants, which are known as gymnosperms, and finally we see the evolution of flowering plants, which are known as angiosperms. The endosymbiosis theory states that a large prokaryote swallowed up a smaller prokaryote with the ability to make ATP. Over time, they developed a symbiotic relationship, and this allowed for the evolution of the mitochondria. The same thing essentially happened with chloroplasts, and so in plant cells, you'll actually see a secondary endosymbiosis where after the mitochondria was taken up, the chloroplast was also taken up. Eventually, we saw the evolution of a bunch of different animal groups over time. This figure to the right is obviously a lot more detailed than you'll need to know, but I'll be talking about some of these groups in the next few slides. One major group that you should know are the eumetazoans. And so this group had gastrulation, which is the formation of a pore. They had a nervous system, and this is where radial symmetry evolved. So a good example of radial symmetry is a starfish, for example. So another key development in animal evolution is the evolution of bilateral symmetry and cephalization. So bilateral symmetry established an anterior-posterior axis, and it allowed for directional movement, and it also allowed for the evolution of a third layer called the mesoderm. And cephalization essentially allowed for the development of a head. The evolution of a mesoderm allowed for the evolution of a coelom as well, and a coelom is a body cavity that develops within the mesoderm, and it allows for better organ support and better digestion. And this is something that's really conserved in animal evolution, since you even see coeloms in us. All animals that evolved after gastrulation are divided into either protostomes or deuterostomes. And so protostomes are where the gastrulation becomes the mouth, and deuterostomes are where the gastrulation becomes the anus, and humans are actually considered deuterostomes. As we move on to chordate evolution, you see much more complex structures, one of which is the notochord, and this allows for structural support and is the most conserved structure within the chordates. You also see a dorsal hollow nerve cord, pharyngeal slits, which were originally for feeding and then evolved to be for breathing. You see a muscular tail, which can be used for propulsion, and chordates usually have a pretty high energy lifestyle. As we move on further to vertebrate evolution, you again see the evolution of more complex structures. So now we have a vertebral column. We have extreme cephalization, which allows us to protect the brain. We have pretty extraordinary sense organs. We have a closed circulatory system, and our internal organs are actually suspended in the coelom. Eventually, we got the appearance of the first hominids, which we believe appeared roughly 4 million years ago. Looking back at the fossil record, we can say that they were already fully bipedal, but they had a pretty small brain size. Despite this, they were still able to develop some early stone tools. Eventually, we moved to middle hominid evolution, where we saw the evolution of the first Homo species, which appeared roughly 2 million years ago. These middle hominids had a larger brain that allowed them to develop more complex tools and possibly even discover fire. This eventually leads us to where we are now, which is explained by the out of Africa hypothesis. This hypothesis states that Homo sapiens first migrated out of Africa about 200,000 years ago and were dispersed throughout the world and actually displaced and eliminated a lot of hominid species. Most notably, we know about the Neanderthals.